Hello, SYNGAP land. My name is Michael Graglia, and this is episode 141 of SYNGAP 10, your 10-minute weekly briefing on everything you need to know about SYNGAP1. I've been away for about a week. You'll understand why in a second. But before I jump into this very dense episode, I just want to point out two things. On Thursday, May 23rd at 5:30 Pacific, we are having a meeting with the leadership team to go over volunteer opportunities. A lot of you have very kindly responded to my request to help and offered to volunteer. And so what we're going to do on Thursday is get the whole leadership team together on the screen, and we're going to have a webinar where we go over all the different groups in SRF and all the different volunteer opportunities. Please join us live. Ask all your questions. We are excited to meet with everybody and try to fill some of these important volunteer opportunities. The other thing I want to flag is that MDBR is 3 weeks away. MDBR is $1 million bike ride. The SRF team bikes to raise money. Aaron and Peter Halliburton have been working on this nonstop for years. Peter is actually fundraising for himself right now for the MDBR, and he's got a $5,000 match. So if you were to donate to Peter's thing up to $5,000, Peter will match it. The SRF's team goal is to raise at least $30,000 because when they raise $30,000, Penn matches it, right? So it's an exciting opportunity to get your money matched, and then Penn will use that money to give a grant for SYNGAP research. So everybody wins. Join us on Thursday to talk about volunteer opportunities, support the MDBR. All right, let's jump in. It's all about therapies. The whole reason SRF exists is to make better therapies for our kids. So, and what do those therapies look like? Well, they could be precision genetic therapies or they could be repurposed therapies, and we need them both, right? Last week, I was at Milken, which, if you listen to episode 140, I was like, I'm going to Milken next week. So now that means last week I was at Milken, this week I was at Ultragenics, and in a couple of weeks, I'll be at Bio. These are all very important conferences, and it's useful for me to go to these things to fly the SRF and the SYNGAP flag, but also to learn and to connect with people. The Milken conference was great. It's one of these very, very high-level conferences. I'm very lucky to have received an invitation. It's because I'm part of this Leaders Link program through the Milken Institute, which I applied to because I want to learn how to make SRF better so that it can serve all of our kids. I talked about this in the previous episode. One of the things I was struck by in the Milken meeting as I was sitting in a session, and it was like the head of ALS and Alzheimer's, all these really large neurological diseases, and they were all talking about how they really need biomarkers and endpoints. And I was like, oh my gosh, we need biomarkers. And all these years, people have been saying to me, make sure you got biomarkers and endpoints. And I thought they were really studying the SYNGAP situation. Turns out everybody says this to everybody. We all need better biomarkers and endpoints in neuro. And I'm really proud of the work that SRF is doing on this. If you remember our conference, we had an EEG talk, and there's also a whole other project that many of us have given blood for that we will be using to work on biomarkers and endpoints. So anyway, Milken was great. This week at Ultragenics was exciting because I was there as a speaker, but I was able to secure an invitation for Kathryn, our CSO, and she was just having the time of her life. She was running around, talking to people, learning things, connecting dots. It was amazing. We're very lucky to have Kathryn on our team. And then Bio, Kathryn and I will be at Bio in a few weeks, which is a huge industry convention where we can really both connect with everybody who's working on SYNGAP1 related disorders, but also hopefully talk some other people into it. Super exciting. Lots of progress on the genetic therapy front. And also last week was ASGCT, the American Society for Genetic and Cell Therapies. And this is a really massive conference where people drop news. And there was news dropped for Angelman, Capsida and Encoded talked about STX, and FoxG1 was running around doing a lot of good work because they're actually developing their own gene therapy because unlike us, they don't have a lot of industry people developing a therapy for you. So they're doing it themselves, which is something I've always wondered about. Should we be developing our own therapies? And the answer is, so far, no. We got a lot of companies working on it. So links to the stuff on Angelman and STX in the show notes. Why do we care about that, Mike? It's not SYNGAP. Well, it's not SYNGAP, but all these companies could also work on SYNGAP. And the progress they make in Angelman and STX will embolden them to go after other logical targets. And we are one of those logical targets, right? Angelman, STX, these guys are ahead of us, frankly. They got their act together faster. The patient advocacy group was in place before SRF was in place, and they have done great work. So we just have to respect their progress and hope that we can be a half step behind them. And if you listen to Nasha Fitter, as many of you know, is somebody I really respect and I follow what she does. She gave a very brief, like 1.5 minute summary of what she learned at ASGCT. And one of the things she said was, this is really the time for precision therapy. So I think we should be hopeful. And I think it's going to take too darn long, right? I think best case scenario, any number of the people on our pipeline identify a target this year. They do the necessary other lab work and talks. And maybe they do a phase 1/2 in 2025. I mean, that would be kind of amazing if they did a phase 1/2 in 2025, and it goes really well. And then they do a phase 3 in 2026, and that gets marketing approval in 27. So even the best case scenario, there will be a marketed drug for SYNGAP1 in like 3 years, and that is like very optimistic. Now, there's be a phase 1 trial before that and a phase 2 trial before that, and maybe we could take part. Or maybe not. Who knows? But this brings me to my next point, which is, in addition to precision genetic therapies, we also need to be thinking about repurposed drugs, right? Why repurposed drugs? Well, because repurposed drugs can help us now. By definition, repurposed drugs are already on the market. They are already available. They are already safe. They are already in humans. And if we find a drug that we think could help Syngapians, we should be all over that, especially since we have years to wait, maybe 2 years before there's a phase 3 trial, maybe 3 years before there's a marketed drug. Maybe. I'm making up numbers. Who knows the future? But unfortunately, the more I think about the regulatory pathways, the more I understand them, I think we're 2 or 3 years out. So this is why I think we also need to focus on repurposed drugs. And I got to tell you guys, there's one researcher in our community who sat me down when we started this, and he said, don't worry about repurposed drugs. Got a big NIH grant. I'm all over it. That was 6 years ago. Guess what? No publications, no repurposed drugs. I don't know what that person was thinking or why on earth they have no sense of urgency, but I figured this out about 2 years ago, and SRF started working at that point really hard on repurposed drugs. And as you know, and as I've talked about before, we worked with Rarebase, now known as Transcriptibio, and Dr. Clement Chow at Utah, and we both of them identified a drug. So before I give you a quick update on those drugs, and I'm going to be quick because there's a lot coming. I just want to remind everybody why repurposed drugs are so important, right? Reason number one, they are cost-effective and globally available. What does that mean, cost-effective and globally available? Well, let's be honest, guys. These precision genetic therapies that everyone's working on are not going to be super cheap, right? They're going to be quite expensive, and those of us in the States are going to have to work with our insurance companies to get them funded. Same in Europe. But SYNGAP doesn't discriminate. There's Syngapians in all kinds of countries, rich countries, poor countries, middle-income countries, etc. And some of these countries, they're not going to have these therapies. Forget 2 or 3 years. We're talking 5, 6, 10, God knows how many years. So if we can find a small molecule, an existing drug on the shelf that can help those kids, we almost have a moral imperative to figure that out, because there are Syngapians all over this planet, and we need to help all of them. So that's one, cost-effective, globally available. 2, these drugs are here now, and people are suffering right now, right? So we cannot wait for precision therapies to finally be marketed when these incredibly smart people at these incredibly stealthy companies finally get through all the hoops that the FDA and other regulators have laid out for them. These drugs are here right now. The molecules I'm going to talk about exist today. They're on the shelf. And kids are suffering right now. Kids are seizing. Kids are constipated. Kids are beating up their family. Kids are getting kicked out of school. Parents aren't sleeping. Yada, yada, yada. We have to act now. And I think another reason to think about repurposed drugs is you got to remember, in medicine, we've generally taken people who were well, got sick, and made them better, right? But we knew what better looked like because they were well in the first place. Our kids, unfortunately, were born sick. They've been sick since the day they were born. And as their brains got bigger and the symptoms became clearer, we figured out they were sick. So now we're talking about taking people who were born sick and making them better. It's something we have much less experience with. And that creates the problem of biomarkers and endpoints. Remember what I said at the top, right? Everyone's worried about, okay, what do we measure? How do we know this drug is working? That's going to be a major issue with these very expensive precision therapies. And it's something these companies are already worrying about. But guess what? If we find small molecules that we can put in Syngapians now and can start to improve their condition, those small molecules could help us understand what a person born sick getting better looks like, specifically a Syngapian. And that could inform the trial design, because remember, drugs fail for one of two reasons. The drug didn't work, or the trial was poorly designed and the drug did work, and even though it worked, we didn't measure the right things, and so the drug didn't get approved. So if we can get these repurposed drugs to tell us what a Syngapian getting better looks like, when the gene therapies come along, we're going to give them a better shot by giving them a better trial. One of the researchers I've been talking to about all of this repurposed stuff, because repurposed stuff has really been getting a lot of attention lately. They said, Mike, I don't know if you guys should be doing repurposed drugs. They said, this might compromise other trials. I said, what on earth are you talking about? They said, well, if people are already using repurposed drugs, then we might not have enough patients for other trials. I found this to be one of the most disappointing and disconnected comments I've heard in a very long time. First of all, asking a patient to not use something that is safe and available today to relieve their suffering so that we can test something that might be safe and might be available in a year or two or three signals to me that you really don't understand how much these patients and their families are suffering. I would argue it's unethical to even go there. But beyond that, and beyond that, we are seeing more diagnoses every single week. The notion that we're going to be so successful with the repurposed drug that we're going to get enough patients on those drugs to compromise these other trials is kind of bonkers. And honestly, I think the patients who will be using repurposed drugs and the patients who you really want in these gene therapy trials are actually different populations, right? The gene therapy trials, they want kids as young as possible to have the longest possible impact. And they want these kids before puberty, because once you hit puberty, the hormones get in and everything's all over the place. So they really want young patients. Whereas people on repurposed drugs generally tend to be a little older, and they've got doctors who are willing to do these repurposing things, and that generally is an older patient population. I don't think that argument is 100% tight, but it is directionally correct. But I think one of the most important points to make in response to this is that, my son's on a repurposed drug right now. I'm giving him 4 pills a day, and I don't love it. And I don't love the idea he'll be on those 4 pills a day for the rest of his life, or 5 pills as he gets bigger, right? This is true for the repurposed drugs and the anti-seizure drugs. But think about if you offered me an ASO where I can give him a shot every 3 or 4 months. Think about Tango with Stoke. Or if you offer me a gene therapy where I can give him one intervention and it changes his brain for his whole life. Do you really think I'm going to be like, oh no, I'd rather stay with my repurposed drug? What are you talking about? Oh, well, if you're on a repurposed drug, then maybe you won't get off the repurposed drug and you won't do the trial, or we won't let you on the trial because you're on the repurposed drug. Probably we'll stop the repurposed drug at that point, guys. Honestly, if we find a repurposed drug that makes it so that we're like, nah, we don't need a gene therapy, hallelujah. I don't see it happening. I think at the end of the day, to help these kids, we got to get into their brains and fix their little brains. But if in the meantime, we have a small molecule right now on the shelf in America, in France, in somewhere, and we suspect it helps Syngapians, shame on us if we don't try. Shame on us if we don't try. So repurposed drugs are important, and I think we have an ethical obligation to pursue them. I'm not talking about an opportunity here. I'm talking about an ethical obligation to relieve the suffering of these kids. So let's talk about the repurposed drugs that we've got. I've got 3 things that I'm tracking right now. There's a couple of others out there, but these are the big 3. Ravicti. I won't give you the whole Ravicti story. Here's the long story short. It's an obscenely expensive drug. It's an egregiously priced drug. It's for urea cycle disorder. If you have urea cycle disorder and you get this drug, you live. If you don't, you die. Horizon owned this drug. They priced it unethically, in my opinion. Horizon's been bought by Amgen. Amgen now owns this drug, and Amgen gave some of that drug to Dr. Greenspan. Dr. Greenspan is testing some patients on this drug. There's a lot of science as to why SYNGAP is a strong candidate. There's at least 10 patients I should know the number because I recruited them, but there's at least 10 patients under Dr. Grinspan's care getting this drug. Thank you very much, Amgen. TLDR, because it's an open label trial. I mean, people are not keeping secrets here. A number of our most severe patients have had significant seizure reduction. And I'm worried about this drug from the lens of cost, because frankly, once I think these results are published, we're going to have a lot of patients being like, hey, my kid's got a lot of seizures, I want Ravicti. And the insurance company will be like, no way. Now, outside of the US, the pricing is different. And frankly, I'm a little surprised that we don't have more people ex-US asking about Ravicti. And one of the volunteer opportunities, frankly, is for each of these drugs to have a team captain and someone to really become expert on how you would get this drug paid for in different geographies, because this is information that we have from the other groups. Anyway, Ravicti, great on seizures. You have to take a lot of the drug, very expensive. Next drug, nortriptyline. Prescription drug, US, antidepressant, old-fashioned antidepressant called a TCA. Today's doctors don't love it because it's a TCA, which means it has 3 rings, which means it interacts with a lot of stuff, which means they have to be really thoughtful about what other drugs they use. Good news, interacts with a lot of stuff, so does SYNGAP, and it seems to be really helping the one patient who's on it that I know about, who happens to be my son, right? So as soon as I saw this data, I said to my doctor, I'd really like to try this, and he said, okay. So under a doctor's care, with the approval of his institution, Tony is on this drug. It's been really interesting, and we've not been shy about telling people, but it's a prescription drug. There isn't a trial yet. We've been waiting to see how it's going for Tony. A couple of people have gone to their doctors. Doctors have said, let's try it. Their doctors have talked to my doctor. Hopefully we'll get a few more kids on nortriptyline. Hopefully we'll see similar improvements in behavior and aggression and other things. And maybe there's a there. Good news about nortriptyline, dirt cheap, available everywhere. Try it. Third drug, acetyleucine. This is not a drug. I should call it a nutraceutical, because you don't need a prescription for this drug. You can just walk into a pharmacy and buy this drug. You can buy a lot of it. You can even mail order it from a pharmacy, which you would have to do if you're not in France, because it's only available in France. It's only available in France. We published a blog on this, a lot of great information, really set off a lot of discussion. A lot of questions from our scientists, our clinicians, etc. One of those questions was, hey, what's the mechanism of action? Maybe we should know the mechanism of action before we try the drug, which really underscores how researchers privilege understanding over urgency and parents privilege helping their kids over trivia. I'm being a little provocative, but let's be honest. We don't know the mechanism of action of many of the drugs that we are using today. See Ravicti. No one's quite sure why that works. We're running a trial on it. Half of the anti-epilepsy drugs we're taking, people aren't 100% sure why they work. A lot of drugs, we don't understand the mechanism of action. So to take a drug like N-acetyleucine that we've been dosing in humans for 60 years in France, and say maybe we should wait and understand the mechanism of action, really privileges, let's do some more lab work over let's see if this really helps kids. And anyway, I go off topic. So on the mechanism of action point, we are about to publish a blog. We have scoured the literature, the science team at SRF in partnership with some scientists and other patient advocacy groups. Because by the way, guys, this isn't a SYNGAP thing. This seems to help kids with broken brains. And at this point, I've lost track, but kids with at least half a dozen monogenic diseases are taking this drug as a result of this blog that we put out. It's kind of remarkable. And TLDR, we're going to put out update number 2 with the mechanism of action, and we're going to put out update number 3 with some case studies, and we'll be talking to our SAB at the end of the month. This is a super interesting drug, and you can just order it from France. People have been like, where do I order it? Somebody sent me a link, bit.ly/Tanganil24. It's just 3 of the online pharmacies you can get it at. I'm not telling you to get it. I'm not telling you not to get it. I'm letting you know this has helped a lot of people. And if you are going to use it, I'm not encouraging you to use it, but if you're going to use it, please connect with SRF and make sure that you track any results with the same forms and rigor that we are tracking everyone else's, because we're trying to gather that data and in a systematic manner so we can read it back to our scientists and maybe design a clinical trial, because I really think this merits a trial. I'll give you a for instance of one thing we've seen, and this isn't a conclusion, but it's the value of coordinating and communicating and collecting data. We've seen a few kids on one anti-seizure drug seem to not respond. And that's been a finding, right? These are very different kids, 3 different genotypes, and yet all 3 of them were like, nothing on this drug. And then we're like, well, what other drugs are they taking? And the smoking gun is this one drug that they're all taking. So more data on that, and I don't want to corrupt the data by announcing it here, but super interesting. So there's 3 repurposed drugs, guys. Acetyleucine, cheap and affordable, available mail order in France. Nortriptyline, available pretty much everywhere, cheap and affordable. Ravicti, stupid expensive, but we already have seen it reduce seizures radically in SYNGAP patients and SLC patients and STX patients. And I argue that we have an ethical obligation as the people with the grace of a diagnosis to figure this out so that we can minimize the suffering for as many people as possible around the world while we're waiting for these genetic therapies, which are almost certainly better when they get here, but they're not here yet. And kids are suffering now. So I think we need to stop saying this is better than that and let's wait, and we need to embrace the mantra of the SYNGAP Research Fund, which is transparency, collaboration, urgency. Transparency, I'm sitting here right now telling you everything I'm thinking. Collaboration, we are working hand in glove with multiple other rare disease groups, and we are gathering data, and we are thinking hard about Ravicti, about acetyleucine, nortriptyline, not yet. I think that might be SYNGAP specific, but that's coming, guys. There's only so much we can do. We need more volunteers. Join us on Thursday. And urgency. Kids are suffering. Get in here, guys. We got to do what we can to help all the SYNGAPians we know and love and all the ones we have yet to meet. As a reminder, I'm done now. As a reminder, newly diagnosed, we have resources on our site, syngap.fund/resources. Social media matters, guys. Ratings matter. Please, on this podcast, you're listening to Apple or on Spotify or whatever, go give it 5 stars. Tell your friends about it. SRF invests a lot of time and effort in these podcasts so that we can reach as many families as possible. Give this podcast 5 stars on Apple Podcasts. Give it a good review. Tell people they should listen to this so that the next newly diagnosed family finds SRF as fast as possible. Also, YouTube, Twitter, LinkedIn, TikTok. We're everywhere, guys. But you have to follow us. So go to YouTube, follow us. 1,000 people follow SRF on YouTube. We have over 400 videos on YouTube. Check it out. Make sure you're one of the followers. 9,900 followers on Twitter. Now, many of us believe Elon Musk has hopelessly broken Twitter. But hey, there's still people out there, and soon we're going to have 10,000 followers. And that's going to be amazing. LinkedIn, 3,600 followers. Make sure you're one of them. If you're a professional on LinkedIn, follow the SYNGAP Research Fund, and of course TikTok. So anyway, thank you very much for listening. It is a privilege to do this work and to work so hard for all the kids of SYNGAP1 with all the volunteers, and the volunteers are countless, guys. It's an amazing community, and I hope that on Thursday you come to that meeting and you join this community. We are moving the needle for SYNGAP and for patients with SYNGAP. And then MDBR, 3 weeks. Go ahead and click on the link in the show notes. Give Peter a donation. And if you're if anything I've said has triggered something, please reach out, mike@curesyngap1.org. I'm excited to hear from you and to work with you to make the future better for Tony and SYNGAPians all over the world.